Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's Neuroscience Research Podcast. Today is April 23rd, 2026, and I'm talking with Vanesa, Nieto, Estevez, and Parul, Varma. Vanesa and Parul are both assistant professors of research here at UTSA. They're part of a team studying the use of brain organoids to understand genetic diseases that disrupt the development of the brain. And in particular, they've studied childhood genetic epilepsy. So welcome, Vanesa and Parul.
Happy to be here.
Pleasure to be here. Yes.
It's wonderful. And I'm Charlie Wilson. So let's start with some background because there's a lot of relatively new ideas in all of this stuff that maybe not everybody knows. So first, is kind of the starting point is what exactly is a brain organoid and how would we make one if we're setting out to do that?
Okay, so basically what is an organoid in general, not just brain. So now there is like one field that they are trying to make like tissue-like structure in a disc, basically in the lab. So we can have, there are liver, but in our case we are studying brain. So basically what a brain organoid is like a group of cells that they like organize themselves in a similar way that happened in the brain of a human or a mouse.
So, you hear about this, you say, scientists are growing a kidney in a dish or something like that. Yeah. And it's easy to imagine a kidney growing in a dish, but that's not really what it looks like.
It's not necessarily like a brain. So it's basically a not mini version, because that also like mini brains is not the right way to call them, but it's just having like a representation of a brain in the lab. But you cannot imagine that it's like a brain like that.
Right. So we're the reason I sort of beat this question to death is because we're used to growing brain tissue in cell culture or slice cultures of the brain. And those have been around for a long time and everybody kind of understands what they are. And this sounds like kind of like a tissue culture, except something has happened to make it three-dimensional.
Yeah.
So first off, what kind of cell culture is it? And then what is it that happens that makes it three-dimensional?
Yeah. So I think one of the things is, so for example, if you do like a slice of the brain, mouse brain, you can have that slide for certain period of time, but in general, if you want to study human, it's hard to get access to the human brain to get those slides, right? Mm-hmm. That is easier to do it with animal model. But the advantage of the organoid is because we use pluripotent stem cell. So basically the way that we start with this organoid is from pluripotent stem cell that we get from the blood of one person. So it's easy to get access to the starting source. So then we use this pluripotent stem cell. Basically, we use different factors to produce different tissue, in our case, brain. So now the advantage is basically that you can grow that brain of any person that you can get this cell from. In the past, having a tissue from a human, basically you need to wait until that person disease. So you cannot study one disease after basically everything is done. So you cannot study the development of the disease. So that is the advantage.
So I take a stem cell. It has to be a cell that has the right capabilities so that you can sort of deprogram it from whatever it was.
Yes.
And then reprogram it back to be nervous tissue.
Yes.
And then you can grow a tissue culture of that.
Yes.
But it doesn't stop there. Because a tissue culture would be like a monolayer of cells. So what happens that makes
So I think now, as the field evolved, so instead of growing cell, just a monolayer of neuron, for example. So now the cues or the guide that we are using help the cell to like organize themselves in a three-dimensional structure. also adding some extracellular matrix, I think like that.
So
then,
you know the tricks. Somebody had to learn how to turn it into get it to grow into a ball.
yes,
Right.
effectively.
yes.
Right.
So in the past was something more like simple tissue culture. Now the technique they are like moving to more complex structure.
So these things grow into little balls,
Basically,
They do.
irregularly shaped balls,
Yes.
and they're developing. They're like baby brain
Yeah.
tissue in some sense.
In a sense, yes.
And they're getting, they're not baby because they started from an adult.
yes.
But they're somehow been pushed back in the developmental sequence and allowed to re-go.
Yes.
And then you pick them at some time. How do you decide when they're ripe? Like, what are you looking for? Is it size or is it some internal structure or something?
So I think it's depend of the question, right?
Yes, it depends upon the question. Whether we want to study what happens really early on in development or we want to study what happens really later in the development. So based on our question, we pick up the time. And coming back to your point of when they are ripe as in the sense the neurons have really matured or not, or how far can we culture them without them developing a hypoxic core so it depends upon the question.
So what's the limit? I mean, what's to keep me from growing one the size of an apple?
People have tried to
So I
culture
think-
them for a year actually,
So
more
some
than
pe-
years.
Some people, I think the longer that I have seen is almost like 4 or 5 years.
Yes.
But it doesn't mean that 5-year-old organoid is like a 5-year-old brain. It's still really small. So right now the limitation that we have about size is that if they are getting really big, all the nutrient, the oxygen, they cannot go into the center of the organoid. So right now the limitation that we have is that.
They don't
So
have blood
basically,
vessels.
that is
They don't
basically
have
one of
blood
the main
vessels.
problems. That
Biggest
because we don't
caveat.
have
Yes.
blood vessel, the nutrient cannot go in the needle. So basically, they don't grow more just because of that.
So what's a size that you could like just dependably do in the lab?
Right now, they
before-
can get up to 1-2 millimeter. So you can see them just without any type of microscope or.
They're like lentil-sized
Yes.
balls, you can imagine.
Yeah.
A small lentil. Yes.
And then what's their internal structure? I want to ask you because I know the answer to that question is different depending on what you've done to the organoid. But if you guys are interested in the cerebral cortex, so you somehow direct your tissue to express the genes that the cerebral cortex cells would express.
Yes.
And when you do that, you say, oh, I've made a cerebral cortex organoid. Seems fair enough. So the cerebral cortex has this very recognizable classic structure. So what is the internal structure of a cerebral cortex organoid?
So right now, the structure of the organoid, so as you are saying, the cerebral cortex, in general, you have the ventricle, then you have a layer of progenitor, and on top of that, you have the different layer of the different type of neuron, the tip layer and the upper layer neuron. So in the organoid, we can recapitulate more or less that structure, but in the brain, we only had one ventricle. So there is only one unit with that structure. In the case of the organoid right now, most of them, they have more than one ventricle. So you can imagine if you had one ventricle here, another here, then there is a point as they grow that the two structures merge. So as they grow, sometimes they lose a little bit that definition of the structure. So now to prevent that, what the people is trying to push is just to get one of those ventricles. So then they can grow and maintain the
and
structure.
make layers and all that
Yes.
kind of stuff. I see. The one that's got what looks like many ventricles, is it really many ventricles or is it one ventricle that winds around and gets cut many times?
The
and
people
makes-
is trying to see if they are
Yes.
connected, and I think some of them they are.
Uh-huh.
But it's more like a
Uh-huh.
worm.
Yeah.
a wormhole.
Worm, yeah.
Okay, so I have a pretty clear idea, I guess, about the organoid. So I'd like to ask why we would pick this sort of complicated and technically difficult to make and study thing to study specifically childhood developmental disorder.
The beauty of it is that just what you asked in the question, specific childhood developmental disorder, they can be personalized. And that is the power of these organoids. We can make organoids from the iPS cells that like the pluripotent stem cells that have been derived from either the blood or the fibroblast of a particular patient. And we can study the mechanisms that are leading to developmental disorder or epilepsy in a particular patient in the dish. So we are kind of modeling the disease in a dish in a very personalized manner. And one thing that we have not talked about is like we can grow a number of these organoids at the same time. So we are not just growing one organoid from one patient, we are growing hundreds of organoids from one patient. Why that is helpful is because if we circle back for certain therapy, we already are establishing a high throughput screening system for therapy. So that is why we are choosing these organoid models to study a developmental disorder, along with the fact that they are human specific, they have human specific cell types, and they're high throughput, and they are personalized.
And I guess it's really the disorder you're interested in. It's not some model that you've created by making a transgenic mouse or something like
Yes.
that, which may or may not be really accurate representation.
And also the problem, not the problem, but the limit of some mouse model that is not easy to make exactly the same mutation that the patient has.
Yeah.
So some people just knock out the gene, but that maybe is not exactly what the phenotype is in the human. So just being able to have a specific mutation that also help to understand these disorders.
So what specific disorder are you most interested in?
So right now, the disorder that we are like analyzing, because they are also the easiest to model in organoid, are genetic disorder. Most of the time they produce epilepsy, but also some other neurodevelopmental disorder. They can have also some of the patient intellectual disability or other condition. So we are specifically working in two genes or disorder caused by mutation in two genes. So one of them is ARX, that is the arista-related homeobox, that is a transcription factor that is known that is really important for brain development, but still had been used or studied for 20, 30 years, and still these children, they don't have an efficient treatment. So that is like a sign that we need a different model to try to understand the disorder.
We don't even know how the gene causes the disease. I mean, one of the problems with epilepsy is if we find a genetic form of epilepsy, it's often a gene that we don't know very much about.
Yes.
And so here we have a disease that we don't know very much about that causes a change in electrical activity in the brain that we don't know very much about. And it's caused by a gene that we don't know very much about. And as we add on more and more things that we don't know very much about, but which are really important, it just starts to look more and more desperate.
Yes.
And also because,
Absolutely.
for example, many of the patients with the same mutation, they don't have exactly the same disorder. So that is also telling us that it's not just one mutation, one gene, one phenotype. There is many other things contributed to that disease.
Probably the background of the person, the genotype background, is also affecting how this disease is manifesting because of the mutation in that gene.
Or maybe even things that you have been exposed in your life and also contributed.
So since you're working with, you can be working just with one patient. You can be saying, this may not, what I find out may not necessarily apply to everyone, but I know it's this person's disease.
But this is why we also need to have like different patients just to be able to conclude that yes, because all of these lines share the same phenotype, that probably is due to the mutation, you know.
So in this, like the one you were talking about, ATX, we don't really know very much about what that gene does. If I went home to my mom and I said, oh, I'm working on this cool gene, ATX, and she said, what did it do? I think I would be kind of hard pressed to say what it does. Like if it was a motor molecule that was moving things up and down the microtubules or something, I could say that, but it's not that. If it was an ion channel, I can say, well, it's an ion channel. So what does this mysterious gene do as far as we know?
It's a transcription factor. So it's a transcription factor that is important, that has been shown that it is important for specification of interneuron lineage. And it is also present, so like if I'm thinking I'm talking to my mom, I would say like there are excitatory and inhibitory neurons in our brain, and ARX is actually a protein molecule that is important to create these excitatory and inhibitory neurons, to differentiate from their progenitors and to help them have the correct balance in the brain so that you don't have epilepsy, or so there's no overexcitation.
And that makes sense because epilepsy, too much excitation, maybe not enough inhibition is like rough, but first order thing you say about it.
But also what complicates this type of disorder, because they are transcription factor. So basically what that means, they regulate the expression of other genes. So then it's one gene, but then affecting, 10, 100 other genes. So knowing exactly what these other genes are doing and all of that is like a cascade,
Yes.
But it must be that there ends up to be too little inhibition, or that the inhibitory cells aren't quite the right kind, or that they don't make quite the right kind of synapses, or they're not excited at the right moment.
Or a little bit of everything.
A little bit of everything, probably, yes.
Yeah.
So what,
Because that is the thing about ion channel, for example, you mentioned. In general, there are epilepsies caused by mutation in ion channel. So at least there is like more specific. There is a channel, it's affecting the neuronal function, whatever. So this is why you have the hyperactivity. But in the case of ARX, can be like many things and many levels. So this is why it's really complicated to understand every single piece of the puzzle.
So do we know anything about the net effect? Like there's just not enough inhibitory neurons, or they're not the right kind? Do we know anything?
So right now, based on what we know from mice and also from our work, it's not just that they are not there. So maybe there are less interneuron, maybe also affecting a little bit more the excitatory, so they can be more or less, but also the interneuron, they don't migrate properly, so maybe they don't integrate properly in the circuit. So all of that, so it's a little bit of everything. So that is also why it's difficult to treat this disorder, because it's not just one thing.
Good. So in your preparation, you can study all of these steps, right? So there's the specification of the cell type, and then there's the cell type migrating into its correct place.
Right.
Yeah.
And both of those are developmental processes. So there's not just one moment at which you're studying that, but it's dragged out over time. And then there's the establishing synaptic connections with the right targets. And interneurons in the cortex are famously many different kinds that makes different kind of synaptic arrangements on different kind of cortical neurons.
Correct.
So in principle, you could have one kind of interneuron that's just fine, and then the next one isn't. And
it doesn't mean that all the interneuron will be wrong.
Yeah.
right.
Maybe some of them they are fine, but there are others that they are not.
So how much do we know about
So
all
the
of
good
that?
thing about
'Cause
the
that- you guys
organoid
have studied
and the
it.
model, we can isolate all of this process. And it's like, okay, let's see if the beginning, like the proliferation and all of that is fine or not. Then we can say, okay, yes, that area is fine or not. Maybe there are more progenitor. Now, let's check if the interneuron, they are differentiating properly. Or now they are migrating. So we can really have different windows during development that we can study and see. And right now, ARX like affect all of them.
Everything.
Everything.
Yeah.
So how about a little summary? Let's start with specification of the GABA neurons. It's a complicated story anyway, because that happens in a different part of the brain. So would you mind going ahead and like telling us about?
So what we know so far, so we have studied ARX in 2 regions of the brain. So in the cortex, that there they are produced the excitatory neuron, so the one that they are like not more active, but in general, like they are more in the brain, in the cortex. So in that area, ARX is producing more progenitor of that. So basically the first step during the differentiation. But we know there are more progenitor, but later we don't see more neurons. So it's possible that this progenitor just die in the process.
This is any old neuron, or is this a specific type of neuron?
The cortical neuron. So the
Okay.
excitatory
So
neuron.
presumably all the
glutamatergic
different
excitatory. Yes.
So because it's affecting the progenitor, probably that will affect all the neuron later. So then the other type of neuron in the brain, they are coming from a different region, that they are the ganglion teminans. So those are the interneurons that we have been talking about, and they are the inhibitory neuron. So that cells, they are formed in that region in the ganglion teminans, and they migrate to the cortex. So what we have seen about this population is that they are produced in a smaller percentage when there is this ARX mutation, and also they migrate farther to the cortex. So you can expect that if they are migrating farther, maybe you would see more neuron in the cortex, more interneuron. But at least we haven't seen that. So what we don't know yet is that, yes, they are going to the cortex, but maybe because they are not completely developed, or maybe because the cortex is also a little bit affected. Maybe they cannot find the right target, and they are not making connection. This is why maybe later we don't see more interneuron in the cortex. But it doesn't matter if they are there or not. What we see is later the activity in the cortex is hyperactive. So there are more activity. So what we now trying to figure out is how they are making the connection, or what is going on at that level.
So let's see. I'm trying to piece together things that you talked about. Well, one of them is there are fewer of them being made to begin with, the interneurons, but they migrate better.
Yes.
And so
Better or
Faster.
faster
And farther away.
and farther. So that's better. Faster and farther. So it could be a wash. You could say, well, if they balanced each other exactly, or it could be there are fewer ones and they get there okay, but there's just fewer ones. Or also normally the ganglionic imminence isn't just making interneurons for the cortex, but they're making them for many other places. Yes. And so they might be migrating off to somewhere else.
Yeah.
So in the end, if you just pick a developmental time point, would you say, and just counted the number of GABA neurons in the cortex, would it be normal?
So in the mice, there are less interneurons.
But
in our study, also the thing is, in a full brain, you know where the cell will go. It will. You mentioned that at the beginning, that the interneuron in general, one type of interneuron, go to a really specific layer in the cortex. Yeah. But in the organoid, because the structure, I told you that as they grow, sometimes they lose the definition. So that layer, they are not that defined. So it's possible that maybe in the organoid, this is why maybe they keep growing, or they keep migrating, just because they cannot find exactly the layer where they are supposed to go. So it's hard to say in the organoid.
They're just lost sojourners in the cortex.
Could be that they are just there, hugging around.
Because the right layers are not there to provide the migratory cues to these
But anyway, we have the
interneurons.
control, and in the control, they are not migrating that far. But far just in that 15-hour period. Yeah. But far is not that they are going away of the organoid, right?
It's in the organoid, so it's not going to be very far.
Yeah.
Yes.
So you can. What are the possibilities for studying the synaptic wiring and the specification of connections and all that?
No, that you can study the same type of study that you can do in mice. You can do it in the organoid. You can label this interneuron with one virus, and then the excitatory neuron with another virus, and then really take a look to the connection that they are making with different synaptic marker, or you can do patch clamp. Any technique that you can think that have been used in mice, you can use just to really study that.
So what's the next thing that needs to be done? I mean, you kind of pose a paradox. You know that there's less inhibition, or at least there's more activity in general in the organoid after this mutation. I guess we don't know too much about that activity. Some of it could just be interneurons firing really fast, I guess. But there's more activity, but we don't know exactly why there's more activity. But that seems a really important question to me because we're trying to complete this story, right?
Yeah.
From ATX to too much firing in
Yeah,
cortex.
right.
How do we fill in the gap about the electrical part of it?
So right now, what the people in the lab, they are doing, so they are trying to study really the synapses that they are making. If they are making synapses, maybe they are not making any contact.
Uh-huh.
So they are like, or doing patch clan just to see if they are like hyperactive, the interneuron or the excitatory neuron.
So how about just GABA? If you just put, like, one possibility is that postsynaptic neuron doesn't have any GABA receptors.
So that is also, at least in the paper that we just published, so also one limitation of the organoid, they are still really mature. So when we add GABA to the organoid, technically GABA should inhibit, right, the activity. So they didn't respond to GABA.
Huh?
So as you know, GABA during development is excitatory. So based on that experiment adding GABA, what we realized that the organoids, they are not mature enough. So they didn't switch yet to GABA being inhibitory. So
they aren't even doing GABA being excitatory at this point, right?
Yeah.
That's odd, isn't it?
Yeah, so it was surprising when we saw that they are not
Yes.
responding to GABA. So now what the people is doing in the lab, they are also trying to improve the condition media and the culture media just to try to get a more mature organoid that they have already
Sure.
get to that point that GABA is inhibitory. So we also need to knowledge that the organoid that they are still immature.
Yeah.
So we cannot expect what that function like a adult brain because they are not yet at that point, you know.
I know one of the things that people do when they're working across species, and different species develop at different rates, is they try to develop a mapping, like when do the axons from the thalamus grow into the cortex in a mouse?
Yes.
When do they do that in a
Yeah.
monkey?
Human,
When do they do that in a
yes.
human?
Yes.
And when does this kind of response start and that?
Yeah.
So is there a way to somehow map the age of the organoid?
So
Yes.
we are trying also like to age, or not to age, but to map the age of the organoid. But also the thing is, that human cell take longer to grow and to get to a mature stage than a mouse cell. So what we are also realizing that it's not just adding factor to try to accelerate that maturation. It's just human cell take a long time.
Some things just take time.
Yeah,
Yes.
so sometimes we just need to wait until they get there.
And you have to keep them going that
Yes.
long.
So you know that you will have a mature brain in 1 week. From a
But you're
human
hoping it
cell.
won't be 19 years.
Hopefully
Right.
not.
Hopefully not. Yes. Yes.
Yeah.
Okay, so these are kinds of questions I'm interested in. Is like, what is epilepsy and where does it come from? But you're really interested in treatments. So what sort of treatments does ATX suggest? What would a modern therapy designer think of when they're thinking about it?
Ideally, for ARX kind of disease, it would be gene therapy.
Uh-huh.
Yes. It's ARX is complicated in the way, as we have learned in the human system, that it is upregulated with the mutation in cortical cells, and it is downregulated with the mutation in GABAergic cells, right? So one way, many of the developmental epileptic encephalopathies are that sometimes only the wild type is there and the other allele of the gene is mutated, which are called as haploinsufficiencies. But ARX is unique in the way that it is present on the X chromosome and it affects only the males. So we cannot, because its expression is different in two different populations, unless we figure out a way that we can increase or decrease in a cell type specific manner the expression of this protein to come back to rescue the phenotype, I don't think that is very feasible. So the best approach for ARX would be to do a gene therapy in the ideal world.
So the problem is a repeat.
It is the repeat. It is a heavy GC-rich region, which is very prone to mutations. And that is why in the first place that there is a mutation in this gene.
So ideally, you will remove the extra
alanines.
But also one of the limitations, even imagine, so whenever the children start to develop like CSR and all of that, so maybe it's 10 years old. So we have been talking that some of the phenotype is that the cell cannot migrate. So by the age that that child start to have symptoms, the migration already stop it, is done. So even if we correct the gene, there are certain phenotypes that we cannot correct. So at least if we can correct the gene, at least what we can prevent is that the disease continue. You know what I mean?
Yeah.
But it's not that we can go back on time just to put the interneuron in the right place. And
it's going to be extremely hard to completely reverse the disease that these children have, to be honest. What we can do is we can manage. If we are able to achieve that gene therapy, we will be able to slow the progression of the disease. But what damage has already been done, to put it in those words, that damage cannot be rescued, actually.
This is the sad truth about
This is the sad truth. Yes.
So how early could you detect this? Because if you could go back far enough in time, you could.
I think in general, they are diagnosed when the children start to develop symptoms. So they have the first seizure, or then you start to notice some learning disabilities, things like that. So in general, yes, take time. So only in the really severe cases, they start really early. Or imagine that the mother has the mutation, just one copy is mutated. So then if you know that that person already has a boy with that mutation, there is a higher chance that maybe they will have another one. So maybe in that case, they can be diagnosed earlier, not based on the symptom, but maybe get like a sequence and all of that. And know, yes, you have the mutation, or the child has the mutation. So most likely he will also develop the same symptoms.
So what are the chances if a mother has this mutation that the son will have the disease?
I mean, 50%. 50%. You have.
Oh, so it's just dominant. So why not just test mothers?
If you have already a child, yes, I think the mothers will be.
These are also rare mutations.
Yes.
So
I see. So
that's why
you do scans of tests that would all be negative.
Yes.
And then you'd finally find one after enormous trouble.
So the normal trajectory with these kind of children is, what we have learned so far is they start presenting with seizures at the age of 3 or 2 or 5. So some of them are very severe at 2 or 1. So they take it to the neurologist, they start putting them on some medications and things like that. Things don't improve, and then they run to different panels of gene testing, and then they figure out, it's a long process for them to actually diagnose that their epilepsy is because of this particular gene mutation.
Yeah.
It's not a straightforward direct route.
And also sometimes the problem, maybe for ARX there is a little bit more known, but for other genes, so yes, they get to that point, yes, your child has a mutation in whatever gene, and then it's like, but we don't know what is causing really the disease. We know this mutation most likely, but we don't know exactly how everything happened. So then yes, you get to the point that, okay, I know what is causing my child symptoms, but now there is
and then there is a dead end for the families, actually.
Yeah, so sometimes it's really also frustrating for them, because it's like, okay, no one can tell us how or what will happen with our child.
Yeah. And that is why these families have really started believing in the power of iPSCs and organoids. And you won't believe that many of these families invest their own money to generate pluripotent stem cells from their children, with the hope that they can be differentiated into organoids, and you can find a mechanism, or you figure out why these children have epilepsy, and in turn to find the treatment.
That's
amazing, right?
Amazing, yeah.
Yes.
how many different kinds of these developmental epilepsies are there that trace back to different genetic causes?
I think right now there have been identified like
100, more than
150,
100, yes.
something like that. And probably the more we know and the more we sequence, probably there will be more gene involved. But also in each gene, there are different types of mutation. So maybe each mutation affect the function of the gene slightly different.
Yes.
But they look enough alike that
Yes.
they would normally present about the same way to childhood epilepsy.
Yeah.
Oh, that's daunting. It seems like a huge task that you have taken on.
It is.
Well, great. It's one gene at a time then.
One gene at a
so we
time,
need to
yes.
take it little by little.
Yes.
Thank you very much, Parul and Vanessa.
Thank you
That's
for
been
having
really
us.
cool.
Yeah, thank you so much for having us.
This has been Neuroscientists Talk Shop.